In sexually reproducing organisms, meiosis results in the formation of sperm cells and egg cells. Cells that undergo meiosis are diploid with 2 sets of chromosomes. However, these diploid cells, after meiosis, produce haploid cells with 1 set of chromosomes. Later, during the process of fertilization, these haploid cells fuse to form a diploid offspring. Thus, meiosis helps maintain the chromosome number in a species. Meiosis, like mitosis, is also preceded by an interphase stage, during which DNA replicates and each chromosome doubles to consist of 2 identical strands of DNA. Meiosis consists of 2 cell divisions, namely meiosis 1 and 2. Meiosis 1 is divided into 4 stages: prophase 1, metaphase 1, anaphase 1, and telophase 1. Similarly, meiosis 2 is divided into 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. Meiosis 1 halves the number of chromosomes, and meiosis 2 splits the sister chromatids of each chromosome, resulting in 4 haploid cells. Meiosis 1 begins with prophase 1, which is typically more complex and lasts longer compared to the prophase of mitosis. Based on chromosomal behavior, prophase 1 is divided into 5 phases, namely leptotene, zygotene, pachytene, diplotene, and diakinesis. All through the leptotene stage, compaction of chromosomes continues and they gradually become visible under a microscope. As the DNA has already duplicated, each chromosome consists of 2 chromatids. In the next stage, that is, the zygotene stage, homologous chromosomes, one from the paternal set and one from the maternal set, get attracted to each other and form pairs. This pairing is called synapsis. During this process, a complex structure known as a synaptonomal complex is formed. This complex structure consists of a pair of synapsed homologous chromosomes called a tetrad or a bivalent. The zygotene stage is followed by the pachytene stage, during which the bivalent chromosomes are clearly visible as tetrads. Moreover, large protein complexes on the synaptonomal complex called recombination nodules begin to appear. These nodules are believed to be the sites where crossing over occurs. Crossing over is an event unique to meiosis, wherein an exchange of genetic material occurs between the non-sister chromatids of homologous chromosomes. The process of crossing over is initiated by the enzyme recombinase and results in the recombination of generic material between the 2 chromosomes. Crossing over is very significant during the formation of gametes, as it results in new combinations of genes, leading to genetic variation in the offspring. By the end of the pachytene stage, recombination between homologous chromosomes is complete and the chromosomes are linked at the sites of crossing over. The next stage in prophase 1 is the diplotene stage. The beginning of this stage is marked by the dissolution of the synaptonomal complex. Moreover, the recombined homologous chromosomes of the bivalents display a tendency to separate from each other except at the crossover sites. The X-shaped part on the homologous chromosomes that holds the two together once they have crossed over is called a chiasmata. The diplotin stage can last for several months or years in oocytes of certain vertebrates. The final stage in prophase 1 is diakinesis, during which the chiasmata terminates. Moreover, the chromosomes fully condense and become clearly visible, while the meiotic spindle assembles and prepares the homologous chromosomes for separation. This stage represents the transition to metaphase 1 and ends with the disappearance of the nucleolus and disintegration of the nuclear envelope. Now, in metaphase 1, the chromosomes line up along the equatorial plate of the cell and the spindle fibers get attached to the kinetochores of the homologous chromosomes. Metaphase 1 is followed by anaphase 1. In this phase, the chromosomes pull apart from each other along the spindle fibers and move to opposite ends of the cell. The chromosomes are still double-stranded with two sister chromatids connected at their centromeres. This marks the end of anaphase 1, which is then followed by telophase 1. In this phase, the chromosomes are at the poles, while the spindle fibers disintegrate and the nuclear envelope and the nucleolus reappear. Cell division or cytokinesis follows telophase 1 and results in two cells referred to as a dyad. Therefore, meiosis 1, which progresses from prophase 1 to telophase 1, results in the formation of two cells with half the number of chromosomes compared to the original cell.